And we will move on to the next slide. This image, and most of these images come from your book. What this is showing you is the buildup of the chromosomes down the middle. And again, remember this buildup down the middle happens because you have microtubules that are attaching to one chromosome via these kinetochores. So you have a kinetochore on each side of the chromosomal pairs. You have a microtubule attached to one kinetochore and attached to the other, and they're pulling in opposite poles. And that equal pulling in opposite poles or opposite ends of the cell is what allows these chromosomes to line up down the middle. And then the enzyme comes along and breaks down the cohesion between the chromosomes. So if there's no cohesion, no sticking together between the chromosomes, then the microtubules can pull the it's actually more like a reeling it in. They're not pulling, they're reeling it in. They're shortening themselves to pull these chromosomes to opposite poles of this cell. In the meantime, at the two ends, you have those centrosomes, which is where most of these microtubules are originating, what they're coming out of. The centrosomes are also putting out non-kinetochore microtubules. What those are doing are actually elongating or changing the shape of the cell. And you have this whole corona of microtubules called the aster that is creating a unique shape at each pole of this cell. It's still one cytoplasm at this time, but the shape of the cytoplasm is changing because of these non-attached kinetochores, whether they be the long microtubules or the asters. And then the other thing you see here is a microscopic kinetochore, very, very large, where it's not real easy to see. This is the one at the bottom to the left, but what it's showing you a little bit are the fibers coming out of the centromere of two chromosomes that are still attached to each other. And then the next image on the right at the bottom, you're actually seeing those microtubules and you're seeing the chromosomes. This is some pretty exciting microscopic work there. During prophase, so we're going back, we just kept talking about metaphase over and over again, but prior to all of this organization, getting these chromosomes into the middle is what's going on during prometaphase. That's where those microtubules are attaching to the kinetochores. They begin to move until they are lined up in the middle. And what you see here in this image is you see that middle lineup, which gets its own name called the metaphase plate. Gets its own little name called, I mean, not a little name, I'm not trying to belittle it or anything. It is an event to the point where we do look at this and we see that lined up down the middle. And then you have two pores and the two spindle poles, not pores, spindle poles, and then spindle fibers. These are each dyed different colors just for visual, but makes it a nice visual. And then the next phase after metaphase, so prometaphase, you have prophase, prometaphase, metaphase, and then when they start, the chromosomes start separating, then you have anaphase. This is actually an exercise from your book, which is kind of nice. What your book invites you to do is to draw and circle. And what you have, what's been done here is the circling's been happened for you. You have a blow up of the previous image, which is pretty amazing. You have the aster. These are those microtubules that are just creating a different shape to the cell. You have the chromosomes down the middle, the metaphase plate, and then you also have the movement of these chromosomes to opposite poles. There's some excellent video of actually showing this and showing this movement of these chromosomes, this tug of war, this pull to opposite poles. A little bit more about this kinetochore. It is proteins. Remember, this is it's attached to each of the sister chromatids, and they're still in this image that you have down on the right. They're still cohesed to each other, so there's still cohesion there. Then the microtubules are going to attach to opposite sides. It kind of helps you get that picture of what's going on and why that tug of war is taking place. And it's a protein complex, like most things that do work in your body, are protein complexes that are associated with the centromere. That's that waist-like area of the chromosome. In anaphase, those sister chromatids separate along the kinetochrome microtubules to opposite ends of the cell. They tend to shorten by depolymerizing at their ends. So they lengthen by polymerizing, so just adding units to the individual basic unit, and they shorten by taking the units off. And then, as I said, the non-kinetochore microtubules will push and pull against each other, basically to change the shape of the cell, elongate it. Remember, one cell is going to become two. It has to elongate. And then in telophase, the identical daughter nuclei form at opposite ends, and then cytokinesis begins during anaphase or telophase, with and the spindle eventually disassembles. So you have prophase, where the nuclear envelope is dissolved and the chromosomes are condensing. Prometaphase, where all of this microtubule activity is being created and organized and being attached to the kinetochore of the chromosomes. Anaphase, chromosomes are being pulled apart, and more activity with regard to the non-kinetochore microtubules. And then telophase, you have the furrow, this cleavage furrow, or furrow. I guess it's a furrow, not a farrow. Sorry, I didn't mean to shift the pronunciation. And what that is, we have a picture of it coming up, but you have actually almost an elastic band, or it's actually a band of proteins right in the area of that metaphase plate that just pulls a drawstring and separates the cytoplasm of one cell into two. And this image you have down at the bottom is another microscopic image of different parts, again labeled, a cell that is getting ready to turn into two cells, so it's late telophase. It's pretty amazing that they can dye this, and you can see this so clearly. And here in this image, this is where this furrow is, this cleavage furrow. And like I said, you have proteins that are attached to the plasma membrane at the area of the metaphase plate, and they just act like a drawstring. They're actually pulling, going to pull the one cell in until it basically becomes two cells. And that is how cytokinesis takes place. Now, that's in animal cells. In plant cells, that doesn't happen because the two cells have to stay attached to each other. It's a plant cell. So what happens is vessels are created by the Golgi apparatus in the plant cell, and those vesicles then create the building blocks of the cell wall between the two new daughter cells. So it's slightly different in plant cells than animal cells. In here, one of the things that I think in order to get things in your head is to have things repeated. So what you have are microscopic views of each of the phases. So prophase, condensing of the chromosomes, the nuclear envelope is disappearing. Prometaphase, this is when you have the nuclear envelope is gone. You have much happening with regard to the spindle fibers. And then metaphase, that spindle fiber arrangement is complete, meeting down the middle. Anaphase, they're pulling apart. And then telophase, the cleavage of one cell into two. This is also another graphic. And again, if they help you, that's good. If not, then just pass this one by. You will try to identify some of these phases in a lab activity, and so you'll have a little bit of exercise with this. The evolution of this process has been studied, and one of the things that have been studied because they can, it's simple enough, is bacteria, and how bacteria, their cell cycle is understood. We know that bacteria reproduce by binary fission, a type of cell division. And what happens is the DNA of the bacteria, although it's not as sophisticated as ours or complex, it's still, I think your textbook talks about if you take the DNA out of a bacteria and stretch it out, it's about 500 times longer than the bacteria. So it's still pretty complex. But what the bacteria has is two areas that are called origins of replication. And what happens is those origins of replication move to the two opposite ends of this bacteria. And one of the things before this happens is this bacteria inside, it just gets too big for itself. So it knows that it's time to, I mean, just based on size, it's time to divide. So the two copies of the origins of replication, that would be kind of equivalent to our DNA replication, but these two copies of the origins of replication move to opposite poles and then start they start to replicate the rest of the DNA so that it actually causes these well, it causes the DNA to duplicate, but in a different way. So you have just these areas that are directing this replication instead of the DNA actually replicating itself. And then after the replication is finished, the plasma membrane grows inward. It's not so much the same way that ours does, but probably probably something that somewhat looks like it, where you have proteins that are actually causing it to pinch in, and two daughter cells result. So not like human cells or animal cells replicate at all, but certainly you can see how it relates. So the relatability of binary fission and mitosis. And then other types of replication with the dinoflagellates, the nuclear envelope never goes away. What happens is microtubules actually split the DNA down the middle and then separate the two nuclei, and then also separate the two cells. With regard to diatoms, the nuclear envelope still remains intact. You do have a kinetochore, and you do have a replication of DNA, but the DNA just is pulled to separate poles, and then you have the nucleus pinching in. Somewhere then after the nucleus pinches in, the cytoplasm is going to pinch in also. And most eukaryotes have some sort of mitotic replication of soma cells. We have a kinetochore, you have the microtubules. The nuclear envelope disintegrates or goes, it doesn't disintegrate, it disassembles, because then it can reassemble. I think it's one of the things that I left out of the telophase is that nuclear envelope is going to reassemble around the